Hey everyone, we hope you're all holding up okay. Welcome to our animation on the phosphatidylinositol 3 kinase pathway. This PI3K pathway is involved in cellular functions such as cell growth, proliferation, differentiation, motility, survival, and intracellular trafficking, which in turn makes it very much involved in cancer. PI3 kinase is a major positive regulator of the PI3K pathway. Specifically, it is an intracellular lipid kinase that phosphorylates the 3' hydroxyl group of phosphatidylinositols and phosphoinositides. There are 3 classes of PI3Ks, class 1 being the most widely implicated in cancer. Increased activity of this pathway is often associated with tumor progression and resistance to treatment. According to Dr. Lewis Cantley of Beth Israel Deaconess Medical Center, the PI3K pathway is an ancient signaling network that has evolved to control cell growth, primarily by controlling glucose uptake and converting glucose into an anabolic molecule to make proteins, lipids, and DNA. He also says that 80 to 100% of cancers use this mechanism. This pathway begins with the binding of a growth factor onto a receptor called tyrosine kinase. The binding causes dimerization and phosphorylation of the receptor. The next step is to activate PI3K, and there are 3 methods of activation in which different proteins, which are determined by the receptor, can bind to a phosphorylated domain. The first method involves insulin receptor substrate 1, or ERS1, binding to the activated receptor. ERS1 then acts as a binding and activation site for PI3K. The second method involves PI3K binding directly to the phosphorylated receptor on the internal side of the membrane and becoming activated. The third method does not include a receptor site at all. Instead, PI3K could bind to activated GTPase and RAS to become activated. Once PI3K is activated, the third step of the pathway is to form the second messenger and activate AKT, also known as protein kinase B. To do this, active PI3K phosphorylates PIP2, which is a normal membrane component that's anchored by lipids, to active PIP3. PIP3 can then activate AKT. There are 3 main effects that AKT has on this pathway and subsequently on cancer. AKT, or protein kinase B, is best recognized in this pathway as an inhibitor of apoptosis by binding to Bax and hindering it from forming pores in the exterior mitochondrial membrane. AKT is also known to activate protein synthesis in a multi-step protein cascade in which AKT binds to Reb, which activates mTOR. mTOR then activates the translation factor S6K, which activates a ribosome to translate a protein. AKT is also known to inhibit a tumor suppressor by phosphorylating FOXO. The phosphorylated FOXO is then ubiquitinated and destroyed by proteasomes. Typically, FOXO would prevent proliferation. So overall, AKT promotes cell growth while inhibiting cell death, leading to unregulated cell growth and eventually cancer. Okay, so this has been a little explanation on the PI3K pathway. Thanks for watching, and we wish you all the best and hope everyone's staying healthy and safe.